Hello everyone, my name is Eduardo Soto, and I will be talking about my project, which is called The Anti-Inflammatory Potential of Alpha-7 Nicotinic Receptor Silent Agonists in Human Blood Immune Cells. So, alpha-7 nicotinic acetylcholine receptors have been of much interest to us for their ability to modulate in vivo and in vitro models of inflammation. Moreover, this ability is thought to be explained by a process known as desensitization, where the channel remains closed and cannot be reactivated for a prolonged period of time. Desensitization is thought to induce downstream signaling cascades that can modulate pro-inflammatory pathways. Now, the recent development of novel molecules known as silent agonists have been of interest to us for their ability to promote prolonged desensitization. And so the objective of this work was to characterize the extent to which alpha-7 nicotinic receptor selective silent agonists could downregulate LPS-induced inflammatory responses by human blood immune cells. And we looked at cytokine secretion profiles of various pro-inflammatory cytokines, and we looked at phosphoprotein cell signaling events. And to assess for our cytokine secretion profiles, we performed a cytometric beta rate to quantify our cytokines of interest. For the cell signaling pathways, we used the Bioplex Pro cell signaling kit to detect and quantify phosphorylation of signaling proteins. And our results here indicate that the silent agonist PCF3DF was able to downregulate the phosphorylation of pro-inflammatory pathway proteins in PBMCs. Moreover, pretreatment with our silent agonist was able to counteract the LPS-induced secretion of pro-inflammatory cytokines, but did not seem to affect the levels of anti-inflammatory cytokines. Ultimately, these findings show that the silent agonist PCO3DF downregulates the phosphorylation of proteins involved in pro-inflammatory signaling pathways, and that they can significantly reduce the secretion of pro-inflammatory cytokines while not affecting levels of anti-inflammatory cytokines. Overall, our findings show that silent agonists can modulate pro-inflammatory responses in ex vivo models of study. Our results further support the hypothesis that inflammation is modulated by alpha-7 nicotinic receptor desensitization rather than channel opening.